by wind and rain. Little impact of the cryptic pest. Now next is development of BT transgenic crop. So what are the status of BT transgenic crops recently in India and globe? So that we will discuss now. So before discussing on this, let me tell you an interesting mechanism of BT toxicity, how this BT affect the growth and how it kill the different insect and pest. Okay, so try to understand that after BT, when it was discovered, a mechanism of toxicity was not understood. It remained a mystery for the large number of years. But after 1950s, the scientists discovered that these crystal proteins, which are encoded by different cry genes of BT proteins, they form the BT spores. They form BT spores. It was previously observed by the Berliner, which is responsible for the BT toxicity. These BT spores are responsible for the BT toxicity. But nowadays, these crystal proteins are called as cry protein. BT crystals protein are called as cry protein, which exhibit a high degree of target specificity because of their mode of action with insect larvae. The larva insect ingests the BT spores and cry proteins. Larva ingests the different BT spores and cry proteins. Crystal protein. You can take example of suppose we are expressing a BT protein into the insect and pest in the different transgenic plants. We have developed a particular transgenic plant which is expressing the cry proteins or cry genes. Okay, so that insect when feed upon this, when insect feed upon the plants, the proteins which got expressed in the leaf are also ingested by the insect. When the cry protein reaches to the gut, it is partially degraded. It is partially degraded, not completely, and it releases a smaller and potential toxic part of the protein. As we already discussed, that when the protein get expressed immediately, that is a protoxin type. Protoxin means it is in an inactive form and it get activated in the alkaline midgut. But when insect had alkaline midgut, when it go there and just face the acid that alkaline environment, the protoxin get converted into the toxin, or partially you can say degraded. Then it releases a partially toxic part of the protein. But this toxin will only be active if it finds the right matching protein receptor. Then once the protein get inactivated by the partial degradations, then protein will look for their receptors in the insect medulla. If receptor is there, then this protein will interact. If receptor is not there, then this protein will not interact and won't affect the growth of the insect and pest. Okay, when this protein found a suitable receptors in the insect midgut of the large insect, this is the most important aspect of the cryotoxin mechanism. Much in the same way, the certain key will only open a certain lock. Cryproteins, cytoxin can only exert its toxic effect on the particular cell receptors. Consequently, the toxic tends to only impact insect within a particular taxonomy. Once the toxin is bound to its receptor, the toxin recruits the different cryproteins, same, and together they form a hole in the cell membrane. They get interact with the receptors, then they make a hole in the cell membrane, ultimately cause the cells to burst. The cumulative effect of this happening to many cells is the irreversible destructions to the midgut membranes, which compromising the barrier between the body cavity and gut. Without this barrier, BT spores and other native gut bacteria can infiltrate and grow within the nutrient rich of the insect and pest. And what makes the BT such a great candidate for the pesticides and GM application is that which these toxins are highly effective against the insect. They have been shown to safety for the consumption by the members. There has been large number of tests by the EPA Environmental Protection Agency have demonstrated that these proteins, like any other dietary proteins, are very unstable in the acidic stomach environment, like human beings have acidic stomach environment. Then oral toxicity, again, further oral toxicity test was performed which involved giving the mice exceptional high dose of purified toxic BT protein. Show no significant health impact in that. In the year 2001, reassessment of several BT protein was done by the EPA that conclude from these findings that there is a reasonably certain no harm will result from the aggregate exposure to the US populations, which including infant and children's. to the crival avian crival F protein, a genetic material necessary for the productions. Similarly, other conclusions were drawn from the crival AC protein of the BT cotton. Okay, so likewise, there are large number of the research has been done. Similarly, environmental safety aspect of BT was done. BT crop has enormous potential to reduce the use of both synthetic and organic pesticides. Nowadays, that's why these organic pesticides company are debating much and much about this BT toxins. Relying on the BT cotton and BT maize, farmers had decreased the pest control, related cost and increased their yield. The USDA report that generally the BT adoption is associated with the lower insecticidal use in 1998-2007. Okay, so that was showing a large potential of the BT cotton in the uh, world at the globe level. Now the recent status, if we talk about recent status of the BT cotton, BT transgenic crop, you can see the first field trial with genetically engineered plant, which expressing this BT, Bacillus thuringiensis toxins, was conducted in the 1986 with tomato. Means first transgenic plant developed through this BT was tobacco. Okay. Then modify they what they did, they modify the sequence of the criven AB protein and criven AC genes to increase the GC content because this is a bacterial protein. So this bacterial protein has a 80 content rich, but to express these protein into the plant system, they have to optimize the codon system, codon uses of the bacterial gene according to the plant genome. Because see, first we need to understand when we are going to express a bacterial gene into the plant system or any plant gene into the bacterial system. So we have to optimize the codon uses. Codon uses means because this bacteria is a prokaryote system, plant is a eukaryotic system. So eukaryotic system is somewhat different from the prokaryotic system. So what we have to do, we have to optimize the codon uses for, according to the individual organism. If it is a eukaryote, and plant gene directly as such, bacterial gene cannot be as such expression to the plant system. We have to modify some nucleotide sequences according to the plant system that make the plant codon usage optimum according to the plant genome. Okay, because some eukaryotic genes has GC rich sequences. So the expression system of plant will identify this gene which is GC rich sequence. It will easily express that kind of protein. Okay, so we have to modify the sequence of triprotein in order to express this protein into the plant system. Require AC genes to increase the GC content. Using this strong promoter more efficiently, polyadenylation and terminalization signals point to 1%, which increased to over 5% when genes were introduced to the protoplast genome. There is a large number of field trials going on the commercial release of the these transgenic crops. Field trial with BT transgenic tobacco were conducted in the United States and France in 1986. The transgenic potato plant which expressing Cry3A from BT varieties, Tenebrionis, were shown to protect the crop against Colorado potato beetle. There has been a first field trial with Bacillus thuringiensis transgenic cotton was conducted in the US in 1988. 1995, the US Environmental Protection Agency approved the first registration of the BT potato, BT corn and cotton crops. In 1998, the EPA approved an insect-resistant tomato line which expressing criven AC protein. In India, GM cotton released by collaboration of the Monsanto and Mike in 2002. The field trial of BT rice were first conducted in China in 1998. A series of transgenic BT rice line transformed with the modified criven AB, criven AC, and criven AC gene which were assessed in large scale of trials in 2007, approved for the commercial release in November 2009. How BT is eco-friendly, pollinator and butterfly. Honeybee hen et al. 2010, Bailey et al. 2005, monarch leaf. Monarch leaf does not have any effect on the monarch butterfly. Earlier it was case as right now we already discussed about this monarch case. Microorganism and microorganism in the soils. It does not affect the group of microorganism. Mite, earthworm and snails. Castaldini et al. 2005, rhizospheric eubacteria. Communities, mycorrhizal colonization, soil respiration, bacterial communities, mycorrhizal establishment. If you take the example of birds and mammals, We already studied about the impact of Bt maize on mammalians and birds. There is no significant health hazard is associated with this Bt maize against these birds and mammals. Effect of Bt crops through multi-generation in mammals and poultry. Against there is no significant human health hazard associated. Aquatic organism also, Chambers et al. Done in 2010 experiments, they concluded that there is no, because the Bt does not have a it does not sustain for longer period. It get biodegraded by a different microorganisms. So what are the advantages? Decrease the use of pesticides. If we are adopting these Bt transgenic crops, so that definitely we have to use less or reduced pesticides. So it obviously it save our cost of crop productions, saving on fossil fuels, reducing CO2 emissions. Because if we are using different insect pests, they definitely release into the environment different CO2s, increase in the yield. There are certain strategies which are adopted to develop the insecticidal activity of crop protein in different crops. First is serine protease inhibitor is from the plant genes. A genetically modified tobacco plant which express the protease inhibitor fused to truncated carbon AB protein. Chitinase is a larval peritrophic membrane formed cylindrical sheets that divide the gut epithelium from the lumens. Chitinase, larval peritrophic membrane formed cylindrical sheets that divide the gut epithelium from the lumen. The network of chitin embedded in a protein carbohydrate matrix. Cytotoxin, CYT, cytotoxic protein cytotoxin 1AA synergize the toxicity activity of Cry11AA by functioning as a membrane-bound receptor of Cry11AA. Okay, so they are showing a different toxic effect on the different insect and pest. Mutation in the binding receptor, binding region of Cry1AA or in site 1AA affected the specific interactions between these protein and reduced their synergistic effect. Now there is a certain modification has been made in the cry-toxic proteins to make it high efficiency or high effectiveness to increase the effectiveness of this cry protein against different insect and pest. So there is different approach. First is side-directed mutagenesis. So side-directed mutagenesis again, mutations in Cry1AA helix protein was done as the histidine 168 position per histidine जो present whatever the histidine is present at 168 positions that has been removed and a different amino acid has been inserted. So this side-directed mutagenesis increase threefold increase in the toxicity effect against the Manducta sexta larvae. Manducta sexta larvae. So it is a very significant significant achievement by the improvement in the toxicity effect of the Bt proteins. So you can see here in the structure at site 168 position they have different at another arginine 120, alanine 128, 282, lysine 283. Second approach is hybrid toxic proteins, which replace the residues 450 domain of Cry1AA, 450 domain of Cry-AA, 450 to 612, replacing the residue that is present between 450 to 612 of Cry1A, of Cry1A, of Cry1A. So you can see by replacing the residue 450 to 612 domain of domain third of Cry1AA by Cry1AC. Deletion of a small region of the toxins. If you are deleting a small region, amino terminal region from the alpha helix, alpha 1 helix of Cry1A protein resulted in the cry toxin that form oligomer in the absence of cadherin receptors. How insects become resistant? There is a mutation in the cry protein receptors as cadherin. So insect may develop resistance against this cry protein also. How? There is a three different approach. Either mutation in the cry toxin receptor that is present in the insect midgut as a cadherin. If mutation occurs, then what happens because of these mutations? Further, these toxic proteins are not able to identify these cry proteins. Cry protein will not able to interact with this. So insect become develop resistance. Elevated immune systems. If insect is continuously exposed against a particular insectal protein, then slowly The insect body get develop the resistance, develop the immune systems against this protein, develop antibody against these systems. Alteration of the activations. They can alter the protocols, the mechanism by cryotoxin activations in the insect midgut. How we can overcome all these situations? By planting a significant percentage of non-BT plants with the BT crop. That is known as refugee strategy. You can plant non-BT crops. So insect will feed on the non-BT crops. So definitely insect will not have the resistance. Gene stacking of different criteria. Pink balloon resistance to Bolgard cotton Cry1 AC, the transgenic cotton Cry1 AC gene has been introduced. There is no resistance to BT2 variety because this contains two different genes, Cry1 AC and Cry2 AB. So instead of one gene, we can take the two different genes, stacking of the genes. Fusion of two or more toxins also we can take the example. Example here we can take that fusion toxin of Cry1 AB and domain 2 from Cry1 AA. Register both coleoptera and lepidoptera insects. We can express higher expression of BT toxin proteins. Now, if you see the BT crop resistance, the data is 2009. In case of 9 BT crops in global market, long list of the BT crops: maize, cotton, potato, tomato, cotton, maize, rice. That explain different genes there. So this is the overall status of the BT cotton in India. Nowadays, till 2003 to 2021, only the BT cotton crop is cultivated in the India, which increased the yield by 38%. Thank you. Thank you for listening.